really my pleasure to be here with you today. As I wrote, after 2 years of pandemics, it was time to start again. And since Leonardo da Vinci was the master of Renaissance, that's why I denominated these seminars as the Leonardo seminars. So in order to start, you need an outstanding scientist. Despite Vivek is very young, I believe he is one of the most brilliant experts in precision medicine all over the world. You can just track his CV to understand how many things he did in a very short time. Dr. Vivek Subbiah is associate professor in the Department of Investigational Cancer Therapeutics and is the Center Clinical Medical Director in the Clinical Center for Targeted Therapy. And he is also Executive Director of Cancer Medicine Research at the MD Anderson Cancer Network. So I believe he is really an outstanding clinical scientist. He served as the principal investigator in over 50 phase 1 and 2 trials. His research interests are drug development in rare cancers. He has led several first-in-human and practice-changing clinical trials that have led to FDA approval. Just think about the combination of dabrafenib and trametinib for anaplastic thyroid cancer, vemurafenib for Erdheim-Chester disease, lurbinectedin for small cell lung cancer, selpercatinib and prasetinib for RET fusion-positive non-small cell lung cancer and thyroid cancer. And his trial portfolio includes trials that target BRAF, MEK, RET, CDK, WNT, VEGF, mTOR, and many other pathways and many novel immunotherapy combinations. He has published over 250 peer-reviewed in several prestigious journals like New England Journal of Medicine, Journal of Clinical Oncology, JAMA Oncology, Cancer Discovery, Lancet Oncology, Nature Medicine as soon as possible. So it's really my pleasure to introduce Vivek, that represents something a genius in precision medicine. And he will discuss about the art and science of precision medicine oncology paradigm, a modern renaissance through the eyes of Leonardo da Vinci. Please, Dr. Subbiah, the podium is yours.
Thank you so much for the amazing introduction. It's a pleasure and honor to be talking to you all. I want this session to be as interactive as possible, so please stop me anytime to answer questions. So let me move on to the presentation. And once again, I thank the organizing team for their vision and mission to ignite the next generation of researchers. These are my disclosures. I will discuss off-label use in my presentation. So what did Leonardo think about the principle for development of a complete mind? He says, study the art of science, study the science of the art, develop your senses, especially learn how to see, realize that everything connects to everything else. Again, you see the symmetry of the human body as evidenced by the precision drawings of Leonardo. In my brief agenda for today, I'll be talking about precision oncology and personalized medicine, talking about early successes in BRAF and RET and future outlook. So each and every one of you should have a personal vision and mission statement. So as a personal vision and mission statement, I'd like to provide state-of-art research-driven patient care, to collaborate with clinicians, researchers, industry, NIH, FDA, and all stakeholders in the world to increase access to new molecular entities and to expedite discovery of the right drug to the right patient at the right time, and to inspire and ignite the next generation of cancer researchers so that we can end cancer and make cancer history. Right now, the world and as we are engulfed in COVID pandemic, and we are coming away from the Omicron wave in US and multiple areas, but still patients are dying and the pandemic will end only when there are 0 deaths a day. So what do we have to look for in oncology? This year, in January, we saw the publication of new dimensions of hallmark of cancer. So as a next generation of cancer researchers, we have a lot to work for. So you may ask, why do we need new drugs, new approaches, and new tests for treating cancer? Cancer is public enemy number one, was before COVID pandemic and after COVID pandemic. As you can see, even during the COVID wave, the deaths fluctuating. The high rate of cancer deaths has not decreased. It's going to be higher and higher. If we keep the momentum, we will have licked the cancer problem in 1,200 years. So that's why we need each and every one of you to contribute to oncology research and oncology science. So let's take a step back. What can patients expect from approved drugs? So these are the current widely approved drugs. And you can see like agents like gemcitabine, timodar, docetaxel, the survival gain is in the order of months, weeks to months. And single agent response rates are 0%. In fact, the response rates to these widely approved drugs vary from 2 to 20%. So how did we treat advanced and metastatic cancers? It's complex. Cancer is complex. So understanding cancer biology is like understanding Mona Lisa's smile. With the subject's enigmatic expression, the monumentality of the problem of cancer, subtle modeling of forms, and the atmospheric illusionism. Today's reality is that metastatic epithelial cancers remain incurable. Statistical victories with p-value less than 0.05 are hailed as major breakthroughs despite survival gain of mere weeks to months. Enter precision medicine or personalized medicine. What is personalized medicine? The management of a patient's disease or disease predisposition and administration of therapies that convey the maximum effectiveness and safety given a patient's unique genetic and molecular profile. And moving from the current practice of one size fits all in a trial and error method to customizing personalized medicine to offering the right treatment to the right patient at the right time. We have immunotherapy. The immune system recognizes the mutanome. We have mismatched repair gene defects in colorectal and multiple other cancers. We also have the entering of high tumor mutational burden. So we have a host of immunotherapies. We have a knowledge of host of immunotherapy targets beyond PD-1, CTLA-4 like LAG-3, OX40, and we have a lot of kinase targets. So we have the knowledge, we have the drug. As Leonardo says, I've been impressed with the urgency of doing. Knowing is not enough. We must apply. Being willing is not enough. We must do. That's why we need each and every one of you to think about how we can cure cancer. It is possible. So, how do we bring new drugs to patients with cancer, navigating the roadway of funding, space, training, staff, collaborating with industry, academia, NCI, working with advocacy, overcoming the regulatory issues in the context of who's going to pay for it, the insurance, overcoming the electronic medical records, providing compassionate multidisciplinary care so that we can make cancer history. So think about the targets for therapies. In the next set of slides, I'm going to be talking about how we can pick one target at a time and make a difference in precision oncology. So in the next few set of slides, I'll be talking about BRAF, talking about basket trials in oncology, talking about the vemurafenib basket study, also talking about the rare oncology agnostic research, which is a ROAR study. And then I will conclude, towards the end with talking about RET inhibitors in precision oncology. So first you got to know what are basket studies. A basket study enrolls patients who have the same genetic mutation, whether their cancer originated in the lung, breast, colon, liver, or any other organ, then has patients receive all the same novel treatment that targets a specific marker. This strategy highlights a different way when we think about cancers, not primarily based on where they originated, but rather based on the mutation that drive them. So you can see that. So mutation A goes to one basket, mutation C goes to another basket, and so on and so forth. The first prime example was the clinical trial with BRAF. Why BRAF? BRAF is seen in 5 to 10% of all human malignancies, and most common mutation is a BRAF V600E alteration. It is seen across adult and pediatric solid tumors beyond melanoma. So based on this prevalence of BRAF, we designed one of the first basket studies called the WEM basket study, in which patients with BRAF alteration, as long as they had a BRAF any tumor type, with any tumor type, they were enrolled in several baskets. In non-small cell lung cancer, the response rate was 42%. And in Erdheim-Chester disease or Langerhans histiocytosis, the response rate was 43%. In fact, this one of the arms, expansion of one of the arms, led to the FDA approval of vemurafenib for Erdheim-Chester disease that harbor BRAF V600 alteration. Beyond these two tumors, we saw responses in multiple rare tumors like pleomorphic xanthocytoma, anaplastic thyroid cancer. At the same time, we saw 0 responses in colorectal cancer. At that time, there was a Nature Medicine publication that showed that EGFR was an innate mechanism of resistance to BRAF monotherapy inhibition. So we added an arm with vemurafenib plus cetuximab, and we started seeing responses with the combination. So this led to tumor agnostic sensitivity of BRAF V600 mutant non-melanoma solid cancers. But because of the wrench of colorectal cancer, it could not be developed as a monotherapy across multiple tumor types. Science moved on. BRAF and MEK was standard of care in melanoma. Why? The combination led to sustained inhibition for observing more prolonged and more durable anti-tumor effect. It delayed and potentially prevented the development of resistance, and also prevented and delayed the development of hyperproliferative secondary malignancies from monotherapy BRAF inhibition. So as science moved on, we designed this ROAR study, which is the rare oncology agnostic research. The idea was simple: pick 9 tumor types with unmet need that harbor BRAF V600 alteration. The patient would receive the combination of BRAF MEK oral agents. And we picked tumor types like anaplastic thyroid cancer, which, as you know, is one of the most lethal diseases known to us. We found early in the clinical trial that we saw phenomenal responses in these patients. As you know, the median survival for these patients is 6 months, and we saw patients doing well. The CT scans show that the tumors literally melted away, and it was dramatic responses in these patients. So based on this early data, the FDA approved the first ever targeted therapy for anaplastic thyroid cancer. So we were armed with 2 successes in precision oncology based on less than 25 patients. And these were FDA approved so that in the real world, any patient with Erdheim-Chester disease or anaplastic thyroid cancer could have access to these drugs. Recently, we published the update of the dabrafenib and trametinib ROAR basket study, and the investigator assessed responses were 56%. And the updated analysis confirmed the definitive benefit of this agent. We also published the dabrafenib and trametinib in BRAF positive cholangiocarcinomas. And this was recently updated in the NCCN guidelines, showing a 51% response rate. And more recently, last month, we published in Lancet Oncology on the activity in glioblastoma and low-grade and high-grade gliomas. Again, a response rate of 69% in low-grade gliomas and a 33% response rate in high-grade glioma with BRAF V600E alteration. Again, precision oncology is possible even in the toughest tumors like glioblastoma, as long as we can deliver the right treatment to the right patient at the right time. The noblest pleasure is a joy of understanding, as Leonardo da Vinci says. So let's move on to the next part of the presentation, which is RET inhibitors in clinical trials and development. I'll be talking about RET aberrations in oncology, RET alterations in non-small cell lung cancer, RET alterations in thyroid cancer and other cancers, talk about why we needed selective RET inhibitors, and also talk about selpercatinib and experience from pralsetinib as well. RET is a receptor tyrosine kinase required for normal development. Alterations in the RET structure and function can lead to tumorigenesis. Oncogenic RET can be driven by RET fusions or RET mutations. RET dimeric fusions are seen in non-small cell lung cancer and multiple other cancers, including papillary thyroid cancer and pancreatic cancer. And oncogenic RET activating mutations are seen in medullary thyroid cancer. It was identified as an oncogene in 1985, and in 2012, it was identified as an oncogene in non-small cell lung cancer. In 2017, we had the first in human clinical trials, and 2020, we had not just one, but 2 drugs approved. As I said, RET is activated by 2 major mechanisms in cancer, RET fusions and RET mutations. So why did we need new drugs to target RET? Because we know that cabozantinib, vandatinib, lenvatinib, sorafenib, and all the dovitinibs, all the small molecules targeted RET. But the challenge is many of these multi-kinase inhibitors have a greater potency on several off-target kinases across the kinome, such as VEGFR2, which led to potential off-target toxicities like hypertension, hand-foot syndrome, proteinuria, hyperpigmentation. So we needed superheroes for it because many patients, almost quarter of a patients discontinued the drug, and 73% patients had to dose reduce the drug. So we needed superheroes. So why do we need superheroes for it? So let's take the world of non-small cell lung cancer. So when we talk about therapies like osimertinib or ceritinib or alectinib in ALK-rearranged or EGFR mutated lung cancer, the PFS is 18 months, whereas with drugs like vandatinib, cabozantinib, or lenvatinib, the PFS order is 6 months. So patients with RET altered non-small cell lung cancer did not achieve the promise of precision therapy. Let me talk to you about one of the first cases that we treated with a selective RET inhibitor. This is a 49-year-old man with advanced medullary thyroid cancer, status post thyroidectomy with RET sporadic M918T mutation. The patient received sorafenib, cabozantinib, vandatinib, RxDX105, vandatinib plus everolimus as part of clinical studies, and unfortunately progressed. When the patient presented to our phase 1 clinic, each treatment was discontinued for disease progression in the liver with hepatomegaly, large volume ascites, severe fatigue, and markedly decreased performance status. He was cachectic and had 30 bowel movements a day. The molecular analysis of the circulating cell-free DNA isolated from the blood before the vandatinib and everolimus showed a gatekeeper mutation, which is a resistant mutation to the first generation multi-kinase inhibitors. So what would we do next? So the patient had a sporadic and a gatekeeper resistance mutation. We knew from the preclinical studies that LOXO292 or selpercatinib was a potent selective RET inhibitor. After showing high kinome selectivity for RET, it was shown in multiple xenograft models that it selectively inhibited RET and was favorable compared to cabozantinib. In fact, it showed blood-brain barrier penetration as well. We treated this patient with a single patient protocol. And because we did not know the dose, we customized and made an NF1 study starting at 20 milligrams of this drug, experimental drug. And we started at 20. We went to 60, 80, 120, and reached a dose of 160 milligram twice daily. Again, remember this, 160 milligram twice daily, a dose in single patient. Again, after a couple of weeks, there was resolution of diarrhea and pain. Calcitonin and CA became normal. The tumors started reducing and patients started doing well. You can see that dramatic drop in the CA and the calcitonin in this patient. In fact, the calcitonin started coming down, and you see a bump here with the asterisks. In Houston, we have one of the major unprecedented flooding events called Hurricane Harvey. And the patient could not come to our clinic for dosing. So the clinics were closed. Patients couldn't come because there was major flooding in Houston. And the calcitonin bumped, the patient did not feel well. So soon as the flood receded, we could open the clinics, the patient started treatment again, and boom, it started coming down. Also, we showed concurrent decrease in circulating cell-free DNAs in this patient. You can see in this one patient, a dramatic reduction of the target lesions. How did I feel? I felt like on first looking into Chapman's Homer by John Keats. Then I felt like some watcher of the skies when a new planet swims into his ken, or like stout Cortez with eagle eyes, he stared at the Pacific and all his men, looked at each other with wild surmise, silent upon a peak in Darien. The Libretto 001 study, the phase 1 dose escalation study, was continued. And again, the phase 1 study was done from 20 to 40 to 60. And the recommended phase 2 dose that we achieved in that single patient was the same recommended phase 2 dose. And that's also the FDA approved dose. So the trial continued on, and it was published in 2020 and was listed as one of the notable articles of 2020 despite the pandemic. And selpercatinib showed efficacy in both chemo-treated patients in immunotherapy, post-immunotherapy patients, and it was active regardless of any of the prior therapies, including MKIs. In fact, the treatment-naive patients, the response rate was 85%. So based on this data set of 144 patients, the drug received FDA approval. So selpercatinib received USA approval on the basis of an expansion of a phase 1 study for the treatment of patients with metastatic RET fusion positive non-small cell lung cancer for the treatment of RET mutant thyroid cancer in both adult and pediatrics. It was a line agnostic approval. It's been subsequently approved in the European Union and also in multiple geographies. Let's talk about the second RET inhibitor to be approved, the Blue 667 or pralsetinib. This is also a highly kinome selective drug for RET, showing more potent and selective than multikinase inhibitors. Pralsetinib potently inhibits RET alterations and resistant mutations while sparing the VEGFR2. The ARROW first in human clinical trial was designed using Boeing design. Maximum tolerated dose of 400 milligram once daily was reached, and the expansion was designed for multiple arms, including non-small cell lung cancer, all solid tumors, and different thyroid cancers. Let me share with you the anecdote of a patient, one of the first patients to get on this study. This was a 27-year-old young man. He had just a baby. He had a baby born at that time. And, young man, his neck was filled with tumor. And unfortunately, they had to put a trach so that he could breathe. The multikinases were not an option because the tumor engulfed the carotid vessels and internal jugular vein. And radiation was not also considered a safe option, given that he could get tracheoesophageal fistula. So we started this patient at a very low dose at 60 of Blue 667 or pralsetinib. And slow and steady, we increased the dose. And the patient had a dramatic reduction of the tumors. Patient's doing well. And he's taking once daily therapy. And he's continuing on. And his child is in kindergarten. And because of the pandemic, we are doing telemedicine visits. And we are shipping the drugs to him. And he's doing so well. And young man, who's able to live a full life, quality of life with his family. The second patient I'd like to share is a 23-year-old woman with papillary thyroid cancer, sclerosing variant. She presented 6 years ago with symptomatic diffuse lung mets and on supplemental oxygen since diagnosis. She was treated with radioactive iodine therapy. She was on sorafenib, then on lenvatinib. She was initiated on pralsetinib at 400 milligrams. She's always been on oxygen since diagnosis. And she had a dramatic response to therapy. The oxygen could be weaned to room air within 5 months. You see the CT scans closely. So you see the asterisks here. This was the baseline. She was cachectic looking and she had poor nutrition. And steadily the BMI increased after 6 months to 22.3. You can see the increase in subcutaneous tissue and fat deposition in this patient. She remains on treatment after 3 years, started college, completed college, is searching for a job, and reached out to me for recommendation letters. I think these are the stories that keep a drug developer, an oncologist, waking up in the morning and going to work. Pralsetinib continued on to show dramatic responses in non-small cell lung cancer and thyroid cancer, and similarly is now FDA approved for RET positive non-small cell lung cancer and thyroid cancers. It's also EMA approved and approved in multiple geographies. So the RET inhibitor superheroes have arrived. Discovery, preclinical, and rapid clinical validation with registrational studies with FDA approval directly from phase 1 studies shows the power and testament to the power of precision oncology. See the timeline for FDA approval from the RET fusion being discovered in lung cancer in 2012 to first in human study starting in 2020 and in 2017 and FDA approval and EMA approval in 2020. 3 years from first in human. Again, these are unprecedented because in oncology, previously it used to take a decade, more than a decade or two to come up with phase 1, phase 2, phase 3 studies. Again, we need to think like the COVID pandemic. Cancer kills hundreds and thousands of patients. We need to be faster, we need to be smarter, we need to be quicker when we design studies so that we can deliver the right patients the right treatment. As we all know, resistance emerges. Cancers follow Darwin's theory. We identified on-target resistance mechanisms to selpercatinib and pralsetinib as in the solvent front and hinge sites of the RET kinase domain. The identified selpercatinib resistant mutants were cross-resistant to pralsetinib. So we need a second generation RET inhibitor that covers gatekeeper and the new solvent front mutations at the same time. So in the next year or so, hopefully we will have an active agent in the clinic, and several are in clinical and preclinical development now. So what about off-target resistance? We saw NTRK fusion as an acquired resistance mechanism to selpercatinib in RET fusion positive small cell lung cancer. So the emergence of resistance mutations severely impacts the durability of tyrosine kinases in cancer. So the next step is we need to overcome resistance mutations. We need combination therapies earlier in the disease course, and develop second, third, fourth generation TKI inhibitors. What I spoke to you about is just genomics, but we need to have a comprehensive portrait of the tumor if we need to win the war against cancer. Again, if we are serious about cancer, we need a molecular portrait of every patient that walks into the door. We need beyond genomics, metabolomics, transcriptomics, immunogenomics, microbiomics, and proteomics, as akin to the parable of the 6 blind men and women scientists who each touch different part of the elephant, such as the trunk versus the tusk, and have vastly different views of the elephant. Panomic testing is a requisite for comprehensive analysis and may require complex computer algorithms for data integration and computation. In 2020, 2021, and 2022, we are seeing the death rate from cancer in the US decline by 29% from 1991 to 20. It is continuing. Unfortunately, COVID-19 pandemic put a wrench in so many of these advances. The sense of optimism in the oncology community seemed to be short-lived as a wrench was thrown in the machinery called COVID-19. We looked into the determinants of mortality for COVID-19, and cancer is an independent cause of mortality in patients with COVID. An understanding of COVID-19 and cancer heavily relies on the timely dissemination of knowledge arising from the most impacted areas. So with Dr. Curigliano, we published the COVID vaccine guidance for patients with cancer participating in clinical trials, and it was in fact one of the top ranking articles in Nature Reviews Clinical Oncology. So the oncology community is resilient to come together for such noble causes. So we need to restore the balance for cancer care in the COVID era. How can we do that? So the clinical trials have been needing to move from being clinical trial centric to patient centric. Sponsors and investigators can achieve in weeks what was previously thought to require months or years. The pandemic highlighted the humanitarian importance of efficient, robust, and definitive clinical research. There have been innovations in clinical trial operations and clinical trial conduct in the telemedicine, from being trial-centric to patient-centric. The next 1 to 2 years may determine how the clinical trial field moves on in the post-pandemic era. As with the painting of St. Jerome in the Wilderness, which is an unfinished painting, precision medicine and precision oncology is unfinished. Think about globalizing precision medicine. So when we look in the map, Most of the studies emanate from USA and some countries in Europe. Majority of the world lags behind in precision medicine. So we need to globalize precision medicine. We need to think locally and act globally so that we can deliver the right treatment to the right patient at the right time. It should not remain an unfinished painting. We have our tasks now ahead. The future generation for the next decade has work to do. We need to work on targeting the new dimensions of hallmarks of cancer in addition to targeting the old dimensions. We need to think about the undruggables, which are the supervillains. So with KRAS G12C, we thought that the KRAS, the beating heart of cancer, is druggable now. So we need to move beyond KRAS G12C with pan-KRAS inhibitors targeting KRAS G12V, G12D. We need to target P53, the guardian of the genome. We need to target the master regulator MYC. We need beta-catenin, it's with its own destruction complex. So we need each and every one of you as superheroes be charging against the supervillains with targeted therapy, immunotherapy, epigenetic therapy, anti-metabolomic therapy. Again, as with the Last Supper, the treatment of motion and complex display of human emotions, cancer is complex. We need to think about the mastery of the perspective. So when we think about cancer, the current paradigm is one treatment for all cancers. Even in precision oncology, one treatment for a tumor type or one treatment for a specific target. Unless we achieve 100% CR rates, even with the best of best therapies, our work remains unfinished. From the current paradigm of one treatment fits all to a customized therapy, a future paradigm for all patients with cancer. Again, we can think about it in a tumor focused manner or in a tissue agnostic manner. Like HIV, we need a heart therapy for all patients so that we can achieve complete responses in all patients. So in summary, explosion in drug development and costs will change how early trials will be designed. There is a revolution in panomics. It's here and growing. Patients will drive clinical research. Social media search and self-driven molecular testing is going to be the wave of the future. The next 1 to 2 years may determine the fundamental pivots in how medicines are developed, giving each stakeholder an opportunity not only to adapt and adopt, but to shape the future clinical paradigm. As Leonardo says, I have been impressed with the urgency of doing. Knowledge alone is not enough. We must apply. Being willing is not enough. We must do. He also says, it has long since come to my attention that people of accomplishment rarely sat back and let things happen to them. They went out and happened to things. I want each and every one of you, the next generation of oncology researchers, to just think about these two ideas of Leonardo. I thank everyone here. I thank Dr. Curigliano, Edoardo, and team for the invite. It's been a pleasure and honor to talking to you all. Thank everyone. Thank all my colleagues. Thank all the study investigators. Thank all the funding agencies for supporting me. Thank MD Anderson Cancer Center, clinical trial staff, and investigators in worldwide collaborating sites for all the clinical trials. Thank you so much.
Thanks a lot, Vivek. Really a great seminar. So now we have some questions. I invite all the people to ask questions in the chat. And my first question is the following. So you clearly show that after the pandemics, we moved from a clinical trial centric to a patient centric approach to clinical research. And then you mentioned the multi-omics analysis. We have several papers now. So do you believe that with the multi-omic approach, we will be able to personalize better treatment, or we will complicate the algorithm? The next AACR will talk about dissecting complexity of cancer, And there is here in the audience also one of the member of the executive board of AACR, that is Adriana Albini. So can you comment on this?
Absolutely. I think that's a great question. So taking a step back, we can think about lessons learned from the chemotherapy era, lessons learned from infectious diseases, right? With HIV, we are not able to cure HIV with one drug. Even for TB, we needed 4 drugs. And even with 4 drugs, we have resistance mechanisms popping up in TB and HIV. So we needed a pipeline of different drugs to go after that. How did we cure pediatric leukemia? Again, it was a regimen of, a 3-year regimen, right? Complicated algorithm regimen. Like, I think we need to have a deeper understanding of the tumor biology. We need multi-omic testing. I think that will give us a knowledge on what we can target. And ideally, we need to have a pipeline of agents that can be combined. I think that's why we need the next generation researchers to think about these complexities of our current paradigm of how we are going to handle these cancers. Even with the best of the best red targeted therapies, we are not achieving complete responses in all patients. We see co-occurring TP53, we see co-occurring RAS pathway alterations that may impact the ability for the cancer to develop complete responses. So how do we combine these agents? How do we prevent these cancers from developing resistance? I think that's going to be the next decade, and it can be a comprehensive topic for a whole another seminar. Talk about how we can address these multi-omic paradigms, because it's going to be complex. It's going to take a whole village to get together to make this happen.
So my other question is, in the recent wiring diagram that Doug Anahan published without Bob Weinberg. So what's about microenvironment? Which is your personal perspective on this? Do you believe we will be able to target also microenvironment in the future, fibroblasts, macrophages? Which is your personal opinion as a leading scientist in early drug development?
I think this is complex, right? Just like, as I said, like Mona Lisa smile, different people interpret it differently. And I think we can get there. I think we need to target the tumor microenvironment. We have cancers like glioblastoma and pancreatic cancer, which are resistant to therapies, to even the best of the best immunotherapies or targeted therapies or chemotherapies in combination. That's because of the tumor microenvironment and fibroblasts, right? So this is a complex topic. Hopefully, I think we have the knowledge, and we are seeing some of the drugs being developed in this space, and we want to see how these can be translated to the clinic. One thing I got to say is, when we target the tumor microenvironment in the metastatic relapse refractory setting, the cancer is quite complex. It's not a single pathway that drives that. Again, and I'm thinking that we think we can still target these molecular hubs if we identify that. And I don't think so we've identified that to date yet. And I think with the explosion in the artificial intelligence, we are able to think about coming up with common nodes or key hubs in the tumor microenvironment that can be targeted. So I think in the next 5 years to 10 years, we will see more and more drugs going after the tumor microenvironment and macrophages.
So we have several questions. I will start with the first one of Adriana Albini. What about sequence of therapies? For instance, first immunocheckpoint inhibitor followed by anti-angiogenic and vice versa. What do you think about this concept of sequencing? And then I have other 2 questions. Yes.
So I think that's a great question. So let me. So let's see. Can you see my screen, or is it?
Yes, yes, we can see your screen. Yes.
Okay. Can you see the slides?
We see the slides of Leonardo, actually.
All right, I see. Okay, perfect. So I was just going to show some other slide. Let me see. Let me pull it up. So I'm sorry, I'm not able to pull it up, but I can talk about that question on sequence of therapies. I think when we think about simple cancers, I think we can strike cancer in the first minute, first round. When tumors are treatment naive, for instance, even in our RET inhibitor studies, we saw better responses to the drug when the treatment was treatment naive. When we start treating cancers after they get chemotherapy, it becomes complex. And it's a paradox that they respond to even the best targeted therapies when the cancer is complex. So I would say that if it's an oncogene driven tumor, uh, we need to start treating them in the treatment naive setting, especially if it's oncogene driven. And checkpoint blockade works in complex tumors, works essentially in patients who have a high tumor burden or neoantigen. So I think immunotherapy can be reserved for later lines of therapy, especially in those patients that do not respond well to immunotherapy. So I would say targeted therapy earlier in the disease course and immunotherapy later in the disease course when the tumor is more complex, genomically complex. And again, we are seeing anti-angiogenics have a huge comeback in combination with immunotherapy with non-overlapping toxicities. So again, as Dr. Curigliano said, anti-angiogenics do target the tumor microenvironment. I think they are able to open up so that some of the immunotherapies and some of the targeted therapies can work better in this space. I think the next question
is of Vincenzo Nasca. So great lecture with increasing histo-agnostic approvals from basket trial, how to deal with rare cancers, that is your topic, where sometimes these data are often scarce due to rarity of disease, and to correctly sequence these new drugs. This is a very complex question.
I think this is a great question, complex question. Again, let's take for instance, let's take an example of NTRK. NTRK is approved all across all solid tumors, and your question can be formatted in a way that a patient with, say, ameloblastoma comes in with NTRK fusion, and because there was no patient treated with ameloblastoma and NTRK fusion in the study or in the clinical trial, how are we confident, right, that this patient would respond to NTRK targeted therapy? That's a complex question. So, again, here, if the tumor, say, responds, you see a response in over 5 to 6 tumor types, I think there is good enough case for it to get a histology agnostic indication. And that's why we need the real world data and NF1 data to feed in to the clinical trial data in these rare tumor types. So how to, again, we cannot do randomized studies because, NTRK or RET or NRG fusions, they are rare. They are subsets and subsets of rare diseases. And we cannot, in the real world also do randomized studies. So two methods to do these studies is using patients as their own control, trial designs, so that usually what we see with NTRK and all these other direct targeted therapies is that patients who are matched do better than they are with their unmatched therapy. I think to correctly sequence the drugs, again, I'm a firm believer that if the patient has an NTRK or RET fusion, it should be given first line, whatever the cancer type is. And that's my personal opinion here.
Onofrio Olivia, which is your idea about future treatment for central nervous system disease? This is really a high medical need. Can you disclose anything on potential mutations and new agents?
Absolutely. As we all know, gliomas have been very, very challenging, and especially glioblastomas are very, very challenging. And more, there have been so many publications, talking about the molecular classification. We have IDH1 mutations and IDH2 mutations in glioblastoma, but we don't have the best targeted therapies, metabolic targeted therapies for that. They are prognostic and predictive, and the therapeutic efficacy of those drugs are debatable. And even with the best of the best anti-angiogenic therapies, although they are approved, they don't confer major overall survival benefit in these patients. What we've shown in our early study with glioblastoma harboring BRAF V600E is that even in glioblastoma, BRAF V600E alterations are targetable. These patients were enrolled in the study in the relapsed refractory setting when they had no other options. I would say the next study has to be designed in the first line setting, sort of a basket, a molecular match for glioblastoma. We have several targets to go after. One is, of course, BRAF, NTRK, RET. We see all these rare targets in glioblastoma. And in the frontline setting, so that we can see what is a natural history of disease, post these therapies. And one of the other things, number two, is, none of these drugs were designed to be brain penetrant, right? Although we've shown all these BRAF inhibitors to be brain penetrant, they are not, designed to be brain penetrant. So we need to design the next generation of agents which are more brain penetrant, where the brain plasma ratio is equal to 1, right? Because we need to go with a higher dosing of these agents that prohibits us from going to higher dosing, mainly because of the systemic toxicity issues. So the next generation BRAF drugs are coming, the next generation RET drugs are coming that are highly brain penetrant to address these mutations. And in fact, in childhood low-grade gliomas, there have been several studies that are being done in this space, and new drugs in the next 5 years. You're going to be seeing a lot of new drugs coming out in this space, which are highly brain penetrant, and we will see if we can have a better overall survival and PFS with highly brain penetrant drugs.
So you presented very nice data on next generation RET inhibitors. So do you envision in the future the possibility to re-challenge with the same tyrosine kinase inhibitor, modifying the molecule against the new emerging mutation? And which is the role of AI? This is a question of Edoardo Crimini.
Oh, absolutely. I think that's a great question. I think what we have is a clinical challenge and a regulatory challenge, right? As we see with the COVID pandemic, right? People now are understanding more and more oncology better, unfortunately, thanks to COVID pandemic, because we are able to explain it better. Cancer, like COVID-19, develops resistance, right? The primary resistance with multi-kinases was gatekeeper, and selective RET inhibitors were done to overcome the gatekeeper. Now we have the solvent front alterations. So there are drugs that cover the solvent front alterations, but they don't cover the gatekeeper alteration. So we need drugs that cover both the solvent front and gatekeeper alteration. So if you're going to cover both the solvent front and gatekeeper, that is for on target. What about the off target effects? We need better drugs for that. We need a customized treatment for off target. So again, ideally, we need to have a pipeline of agents, right? A pipeline of, say, selective RET inhibitors that have activity against each of these resistance mutations, right? And AI is able to predict what are the resistance mutations when, especially in the 3D domain, when a drug inhibits a specific mutation. So AI can predict that, but we need real clinical sample data to see if that resistance mechanism emerges. So in the next decade, we need to have a pipeline of agents so that we can sequence these alterations in real time and find out if the patient has a specific mutation. We can get that particular drug to that particular patient. One of the main challenges is a regulatory issue. How would a drug for a specific mutation with a very, very rare subset of a tumor, how can that be approved, right? So that's a huge regulatory question. And I'm thinking, again, aloud, that something like emergency use authorization. I think for COVID pandemic, everyone came together, thought that is human emergency. Cancer is human emergency. So I think with such agents, as long as we have some safety, there has to be an emergency use authorization mechanism so that the patients can access these specific drugs.
Another question of Paolo Manca. This is very interesting. So you target the genetic aberration, but sometimes you have transcriptional and translational alteration that may occur after the targeting. How can you manage in the future the post-transcriptional and post-translational alterations?
Combination therapies. I think customized combination therapies would be the way forward. I think we need to see how we can combine these agents. And gone are the days with chemotherapy where we have to do phase 1 studies for every combination. I think we can be, as an oncology field, we can be creative, right? Come up with future NF1 customized combinations for our patients.
There is Samart Sandeep from India. What is the role you think regulatory environments will play in personalized oncology? So do you think places like India with cheaper regulation payments could, like Prof, EU and USA? This is a critical question, access to innovation. For India, that will be a leading country in a few years, I believe.
Absolutely. I think, again, so far, I think FDA has been the forefront of modernizing regulations. They have had and been the role model for the entire world. They've approved all these agents via accelerated approval mechanism without a randomized study. I think that's welcome. And that's a first step. I think each and every country needs to come up with their own methods and ways how to fast track and make it easy for patients to access these therapies and not make it harder. And I think you're right. I think with cheaper regulation payments could definitely, countries like India can leapfrog the EU and USA. Again, we will see. That's why I showed that we need to. The work of precision medicine is unfinished unless it is globalized, and we want to make sure precision medicine can be realized only if every patient in every country gets access to all these therapies. And again, regulatory creativity and regulatory flexibility are needed for this area. Thank you.
So short questions for short answer. The first one, how you envision the role of precision oncology in the context of early detection of cancers?
Oh, absolutely. I think that's another topic for the whole seminar. Again, we are seeing a lot of early cancer detection, right? I think we are able to have liquid biopsy methodology for screening cancers. And again, 5 years ago, they came up, the technology was not as good. I think the technology is getting better and better. And there are some companies that come up with early micro mRNA and micro DNA detection of cancer. And I think, again, when we see cancers, we see in the metastatic relapse refractory setting, and you need a Hercules to cut off all the heads of the Hydra. But here, I think the earlier we detect in stage 0 or stage -1, we can prevent cancers from happening. I think that's where more and more research needs to happen. I think that's a great question. I think more and more research needs to happen in the early detection space.
Thank you, Federica. And now, Matteo, beyond super drugs, do you believe that repurposing old drugs can be another strategy in precision medicine?
Absolutely.
Drug repurposing, yes.
Absolutely. I think drug repurposing can be used in precision medicine. And the challenge is how many drugs have been repurposed. I think we need to think about, again, as I said, we need to have a pipeline of agents, right? For instance, a drug that has been shelved, it's sitting in the shelf, but it may work on the red solvent front. We can easily repurpose that. And our drugs like metformin, right? We know that they are now inhibitors of mTOR, But the challenge is no one wants to develop the drug, mainly because it is off patent and it's a cheap drug, right? And we actually don't know the cancer dose for a drug like metformin. I think drug repurposing would also be a great way, because we may already have drugs that are sitting under our nose. I think the COVID pandemic opened the whole world to drug repurposing, right? We did not have a specific drug in March, April of 2020. In a month, in a couple of months, they had the remdesivir data. It was a repurposed drug. It was something, right? It's not like the Paxlovid or the other drug, that was, but it was something for us to help patients with COVID-19. So I think we are right now, if we think about COVID pandemic for like 2-3 years, we are probably in July 2020 of the COVID pandemic for cancer. We know something, and we have some drugs to play with. I think in that scale, we need to know more with a panomic effort. We need to have super drugs, and we need to see how we can sequence these drugs.
So here I have more questions, because it was really success. You inspired the people. So just a short question. First, how much do you collaborate with basic scientists? Second, what is finally, in your opinion, in a precision oncology, the question is whether you can select a specific target when you have several targets. Finally, what do you think about ADC? And Gabriele is asking finally about what is the role of independent single clinical investigator institution? So beyond the drug supported by pharma, which is the role of academic research. And this is the last question, because we did a lot of questions. Okay?
You need to, it's a teamwork. I think it's a team sport. It's not a single individual effort. If you see any game, right, it's like assembling a basketball team or a football team or a soccer team. You need players. You need multiple players for this. You need to work with basic scientists. You need to work with, collaborators. You need to work with pharma, academia. It's not just one clinical investigator. So the clinical investigator can provide that knowledge and clinical investigation, but you can inspire and feed and work with basic scientists. I think in 2022, gone are the days where a single investigator could do everything all in one person. I think technology is fast moving. We need to work as teams. It's team science. We need to have major teams. For any game, think about it as a game. You need, in every basketball team has, you need to have LeBron James. You need to have a Steph Curry. You need to have people with different skill sets in that team. You need to have a basic scientist. You need to have people with regulatory expertise. You need to have people with statistical expertise. You need to try and work with everyone. Keep your mind open. I think one thing is that no one knows about cancer, right? I think no one can think that everyone knows everything about cancer. You need to have an open mind when you work with basic scientists, when you work with statisticians. And I think as young investigators, you need to read statistics well, and also make friends with statisticians. With a good statistician, you talk to them, I think listen to them, and you go back to them with your problem, right? Again, most of the studies that were done before when I was doing my training were all randomized studies. And I couldn't see why randomized studies should be done. So I used to challenge my statisticians, and I used to even bring them to my clinic. Several of my statisticians, I brought them to clinic. I said, you know what, this is what I'm seeing here. I don't want to give placebo. I don't want to give substandard therapy as an arm to my patient. I come up with different ways of statistics. We are not in the 50s and 60s where we can just throw in some random therapies and keep our fingers crossed. Please come up. These are drugs specifically customized for these oncogene-driven tumors, and see how the patients are doing well. This is a slam dunk. We need to please think about some different statistical methodology. We will name that statistical algorithm with your name. So, we want you to be famous. Please come up with some innovative idea. You work with people, you work with statisticians, you work with basic scientists. Talk to them. He said, hey, I just sequenced the on-target mechanism. I don't know what it is. Can you do single cell sequencing? Can you please help me with a bioinformatics algorithm? Again, you have to work with a multi team to think about this. Even same thing with regulatory flexibility. Hey, this is it. So you pick up the phone, talk to people. Hey, this is what it needs. So we need to come up with a study design so that the regulators are agreeable to the study design. So you need to work. It's a teamwork. A single investigator era is gone. You need to have a multidisciplinary teamwork. It has to be a large team in order to win this battle.
Thanks a lot, Vivek. It has been a great success, better than a Mona Lisa. So many people here listening to you, and it was really a pleasure to have you with us. Really, I appreciate a lot, and I hope to see you in person, okay? Thanks a lot, and thanks to everyone. Thanks.
Thank you so much for the invite, and thank you so much. Yeah, thank you.
Thanks a lot, Vivek. Bye-bye.
Bye.
Oh, people are not leaving. Okay. Thank you. Thanks a lot again. I see you soon in ASCO.